class has been affected by sleeping sickness. It would seem to have a propensity for males. Anyway, let me tell you something that you should be happy with. First of all, we have another 2-hour session scheduled for this subject for Wednesday, and don't need 2 hours. 1 hour will be plenty. So instead of meeting at 8 o'clock as scheduled, make it 9 o'clock. Okay? As I think I told you earlier, I had nothing to do with the construction of the schedule here. It was put together by a committee whose composition I'm not even sure about, which is why I missed the very first lecture in this course. Nobody ever told me that there was the class schedule for that day. Okay. We have, in the past, talked about a number of glands that are all in the class of glands that we call exocrine glands. And you may recall that exocrine glands are glands that secrete their secretory product into a duct, and this duct leads to a body surface. Today we will be talking about endocrine glands, which used to be called ductless glands. As the name would imply, these glands do not have any ducts. That immediately tells you something about the histology of such glands. They consist of epithelial cells that are packed together in various configurations. We'll be talking about that as we go through various glands. And they don't have ducts, which is why originally these organs were called ductless glands. They don't have a duct. So where does their secretion go? Well, whatever it is that they're making is exocytosed into the surrounding fibrous connective tissue, and then it diffuses into the numerous blood capillaries that surround the secretory cells. Okay, so no ducts, highly vascularized. Those are the common characteristics of endocrine glands. Endocrine glands produce secretory products that we call hormones. And hormones are of various biochemical composition depending on the organ that we're talking about. They can be steroidal, they can be polypeptides, protein polysaccharides, and the like. And we'll be mentioning the various ones as we go along. Hormones, in effect, are a way of integrating the bodily functions. Hormones regulate most of the things that our body does. And in this sense, they are companions of the nervous system. And in fact, as we go along, you will see that the nervous system may, in fact, under certain circumstances and certain nerves, are involved in the production of hormones. The first organ that we will be talking about is the pituitary gland. Did you ever wonder where that name pituitary comes from? It's sort of a strange word. it harkens back, it harks back to the ancient Greeks. The Greeks thought that this particular organ was a site where mucus was made. What do you do with mucus? Well, you tend to spit it out. You ever read in the comics, if people still read comic books, when they try to indicate somebody spitting, what is the little sound that they put into the balloons? It's usually tui, P-T-U-I. And that's exactly where pituitary comes from. It refers to the fact that the Greeks thought that the product was something that you spit out. Well, that name was accepted, obviously, for centuries. Then, at the beginning of the 20th century, there was an American urologist, endocrinologist, who was offended by the vulgarity of the origin of that term. And he suggested a new word for the hypothesis. Well, that word met with a certain degree of success, but most people who work in this field use the words pituitary and hypothesis virtually interchangeably. Hypothesis usually is used in compound terms, like a neurohypothesis. Easier to say than neuropituitary gland. So I will probably use the words interchangeably, but they refer to the same organ, irrespective of what name we call it. Now, the pituitary, or hypothesis, if you will, has a dual embryonic origin. Part of it, the epithelial component, actually represents an ingrowth from the roof of the developing mouth, the stomache, so that this is the roof of the mouth. A cord of cells invades the overlying connective tissue, and then finally produces a mass of cells. And this ingrowth is called Rathke's pouch. Now, Rathke's pouch should be of interest to dentists. As I said, this is an ingrowth of the roof of the mouth. And occasionally, there's some embryonic misadventure. Oral structures get picked up by the ingrowing tissue and get carried up into this developing pituitary gland. And foreign tissue frequently is embedded in the glandular portion of the pituitary gland. I remember years ago when I first came to Western Reserve, a man who used to teach pathology showed me a slide, an x-ray, taken by one of his predecessors way back, maybe in the 1940s. And it was an x-ray of the front part of the pituitary gland, and embedded in it, right in the middle of it, smack dab in the middle, was a fully developed molar. tooth right up in the pituitary gland. Well, that's very uncommon that a tooth unlocket would get carried up into the developing pituitary. But very frequently, salivary glands get taken up and become part of the pituitary. Ordinarily, this is meaningless. It's only if that salivary gland tissue should, for some reason, become malignant, which does happen once in a while, and forms a tumor right in the middle of the pituitary, and it makes for a real enigma for pathologists who are trying to arrive at a diagnosis. What is it exactly? Well, as I said, frequently salivary tissue ends up in the part of the pituitary that comes from Rathke's pouch. Anyway, this first part that is developed from Rathke's pouch is very, very epithelial in its appearance. But that's only half of the pituitary. The other half of the pituitary is caused by a downgrowth of tissue from the floor of the brain, from the hypothalamus. Now, I should mention that with the passage of time, Rathke's pouch disappears. So that connection is not present or discernible in an adult, but in a fetus, very obvious. Okay, so what we end up with is a situation where we have an epithelial portion, and then there is a downgrowth from the floor of the hypothalamus. And now we have two distinct divisions of this gland. The part that came from Rathke's pouch has a number of names. It is called the pars anterior.
Dr. Tandler?
Let's leave that. I don't want to make a lot of things. Pars anterior. So called the pars distalis. And the part that came from the hypothalamus is called the pars nervosa. A dual embryonic origin. Two different parts that come together to form a single organ. Now, just to orient you, the optic chiasma is in this direction. Okay, now we can look more carefully at this, and it turns out that there are other parts of the pituitary gland. There is what is probably the remnants of the original Rathke's pouch that runs sort of kind of fuses with the pars anterior. And this is called the pars intermedia. And there is one more part. So it sits like a collar around this extension from the hypothalamus, and that's called the pars. Okay, so there are 4 distinct parts of the front of the pituitary gland, and then
Can you move the sheet a
pars
little
nervosa.
on the screen?
Do you mind moving the sheet?
Oh,
Thank you.
sorry.
Thank you.
Okay, if we look at cells that make up the pars anterior, pars distal, they're typical epithelial cells, and a certain percentage of them have secretory granules that are stable. The remainder have secretory granules that don't stay very well. And it is possible then to distinguish 2 classes of epithelial cells in the pars anterior. These are called chromophils, CHROMO, phil, versus chromophobes. Chromophils are cells that are stainable, just with H&E, and chromophobes are cells that don't stain very well. So in the light microscope, they look almost blank except for their nuclei. Now we now know that the pars anterior produces about 7 different kinds of hormones. And each of these hormones is produced by a different kind of cell. Back in the old days, histologists were very much concerned as to which cell makes which hormone. And they came up with a whole series of different staining combinations that could differentiate between the various cells and thus identify the source of specific hormones. And we don't need to do that anymore. It's very easy to identify the source of any given hormone using a technique called a fluorescent antibody technique. Let me very quickly tell you what that's about. Suppose there was a hormone that you wanted to find out the source for. You isolate the hormone and inject it into another species of animal, like a sheep or whatever. That animal is going to make antibodies against this foreign hormone. You can now isolate the antibodies, and using some chemical tricks, you can link a fluorescent dye to that antibody. Then what you do is you take a slide of pituitary unstained and flood it with this fluorescent antibody. When examined in a fluorescent microscope, a fluorescence microscope uses ultraviolet light as the source of illumination rather than visible light. what will happen will be that the antibody, the fluorescent antibody, will link to the hormone in situ, and the cell that makes that hormone will light up. It's quite amazing. I mean, it's brilliantly lit like neon. And okay, you know that the cell makes that hormone. And you can do that with any given hormone and identify specific cell types. That being the case, we can skip this whole business that I used to have to give a class about the different stain combinations that were used to identify specific cells. However, using this modern technique of fluorescent antibody, you can, without any error, without any problem, identify specific cells that make the different hormones. Okay, this not being a course in endocrinology per se, just let me tell you about the hormones that are made in the adeno, that is the front part of the pituitary. First of all, there's growth hormone. Growth hormone is involved in the growth of bones throughout the body. Our long bones increase in length by continued division of cartilage cells in structures called metaphyses. I remember we sort of skipped the growth of long bones, but just to remind you, if you haven't read about it, our long bones, as we are growing, we have plates of cartilage towards the end of the bone. This cartilage gets thicker and thicker by interstitial growth. As it gets thicker, it pushes the end of the bone outward so that the bone increases in length. However, as the metaphysis is pushing the end of the bone outward, it's being destroyed in part along its bottom. So the metaphysis never gets really thicker, but it's moving the end of the bone upward. The operation of the metaphysis depends largely on growth hormone. Now, eventually, the metaphysis will become insensitive to growth hormone, and by a kind of negative feedback, the cells that are making growth hormone in the pituitary will stop making growth hormone. Now, you may not be aware of the fact that up until a few years ago, every hospital that performed autopsies was constrained to remove the pituitary from the cadaver, irrespective of the cause of death. And they would collect it. And at intervals, a representative of the National Institutes of Health would come around and pick up these collected pituitary glands. These glands were then subjected to appropriate biochemical procedures, and growth hormone was isolated. It was used clinically for children who were showing little natural growth. And it was possible to calculate, was a child going to grow to be at least 5'4" if it was determined that a child would not get to be as tall as 5 feet 4 inches tall, this collected human growth hormone would be injected, and this would permit growth up to a reasonable level. Now, before they started doing this, what would happen would be that the metathesis would lose its sensitivity, but the body would still make growth hormone, which meant that one could not grow body height, but that some bones were still responsive to human growth hormone. And this is what happens in little people, or use the politically incorrect term, midgets. Midgets have short bodies, but the heads, which consist not of long bones, but of flat bones, are of normal size. So growth hormone is, in a sense, is a problem. Now, sometimes an individual will end up with a tumor of the cells that makes growth hormone when they are children. This means that there is an excess of growth hormone, and such individuals grow extremely tall, will probably end up playing professional basketball because the metathesis remains active and is working to beat the band. People really grow very, very tall. Now, suppose the metathesis has stopped working, it's lost its sensitivity, and a tumor occurs in those cells that make growth hormone. Well, what will happen is that the flat bones and certain long bones will respond to the presence of that excess hormone and produce distortion of flat bones, the hands, and so on. And that is the condition called acromegaly, which I'm sure you've heard of. And acromegaly, that is a condition where, because of a tumor, usually in the pars distalis, human growth hormone, is made in excess when there is no cartilage to respond to it, and the flat bones keep growing and produce that distortion that you've probably seen pictures of. Now, it's rather interesting that most hormones are species specific. That is, a hormone that works well in one kind of mammal probably will not work very well in another mammal. Growth hormone is injected into much of our dairy cattle, and all the milk that you drink has growth hormone in it. And this is probably responsible for the fact that Americans are getting taller. I know back in my day when I was in college, the height of the average male freshman entering college in the United States was 5 feet 7 3/8 inches. The height of the average male now entering college in this country is 5'10" and a half. It's like 3 inches more. So it's growth hormone that has been put into our milk that's really responsible for that change in height. Again, low growth hormone are quite specific in terms of human beings. The stuff that's in cows and so on barely has any effect, but it shows. So the increase in the height of both men and women in this country is due largely to growth hormone in our milk supply. Okay, so again, if we look at the PARS interior, it consists of clusters of chromophils and chromophores with a lot of blood vessels in between. If we now move over to the PARS intermediate, that is really still a remnant of Bradley's pouch. You remember the Bradley's pouch had a tubular connection to the outside. Well, the connection is false, but the tubule has some lumen in it, and some bits of that lumen are often preserved in the PARS intermediate, so that if you examine it in a light microscope, you will see that in the PARS intermediate there are some follicles that are present. These follicles sometimes contain a material which is called colloid, not to be mistaken for the material that's found in the thyroid gland, which we'll talk about in a little while. PARS intermedia is, in human beings, not a very clear-cut component. It tends to sort of fuse with the pars distalis in front of it, and the cells sort of become intermingled. We know one hormone that this part of the gland makes, although in human beings it's not very important at all. And this hormone is called melanocyte stimulating hormone. And what it does is it stimulates melanocytes in the skin to make melanin. Now, in human beings, the action of this hormone is barely visible. Probably the best place to see it is during pregnancy. It can cause a darkening of the areola of the nipple of the female breast, so that the color darkens. And it's simply because melanocytes in that region are making melanin. Other than that, it's really a very trivial kind of hormone in human beings. In some animals, it's very important, like in chameleon, for example, which can sort of change color. And that is due to the rapid release of melanocyte-stimulating hormone, which affects the distribution of the melanocytes in the epidermis. And you can even see this with an ordinary frog. If you take a frog, put it on a light background, it has sort of a light skin. And if you take that same frog and put it on a piece of black paper, its skin will darken. And again, it is due to melanocyte-stimulating hormone. Again, in human beings, trivial. Then before we leave the anterior pituitary, I should mention that pars tumoralis is almost a completely unknown part of the pituitary gland. It has, it's probably the most vascular part of the entire gland, but even though we know that it's got all of these blood vessels, it's not clear why. Not even certain exactly what hormones they make. Now, before we leave the pars anterior, that is this front part of the gland, I just want to mention, and it's not important really, some of the hormones that are being produced. There's 7 different hormones there. We talked about growth factor. One that will be of importance for us the next hour is thyroid-stimulating hormone, otherwise known as TSH, thyroid-stimulating hormone. Many of the other hormones have to do with maturation and function of various parts of the reproductive system in both males and females. For example, there's a hormone, prolactin, which is involved in the production of milk during and after pregnancy. And then the maturation of the ovary and the menstrual cycle are in part controlled by pituitary hormone. And the function of the adrenal gland is controlled by a hormone that I'm sure you've heard of, called ACTH. Okay, so the whole series of hormones that are produced here, and don't worry about them except the ones that we've spoken about in detail. The most important one will come into our discussions this time and next time. Let's move over to the posterior lobe. Now, as I told you, that part of the pituitary is basically a downgrowth of the hypothalamus. And if one examines it in a light microscope, you see that it consists entirely of axons, completely consists of axons. And cell bodies are back up in the hypothalamus. These cells produce hormones, these cell bodies, and there are 2 kinds of hormones that are produced here. One we've already talked about in connection with the kidney, and that hormone is anti-diuretic hormone, ADH, which has a bunch of old names like pitressin and vasopressin, which you probably heard back in college. ADH is the commonly used abbreviation for it. You may recall what the function of ADH is. In the kidney, remember that after the initial urine has gone through many yards shards of kidney tubules. It ends up in the collecting tubules, and it's still pretty dilute. And the ADH keeps the collecting tubules permeable to water, and water is withdrawn from the collecting tubules so that urine reaches its final concentration. In the absence of ADH, or with the deficiency of ADH, the individual produces huge amounts of urine in a condition which is known as diabetes insipidus, to be distinguished from diabetes mellitus, which is the sugar diabetes. Diabetes insipidus, an individual can produce 25 liters of urine a day. I mean, that's an enormous amount of water. And it's all based on the absence of ADH. So that's a hormone that is made in the neural hypothalamus. The other hormone is a very interesting one called oxytocin. Oxytocin. Now, oxytocin was first discovered as a hormone that causes myoepithelial cells to contract. Now, you remember what myoepithelial cells are. These are cells that are sort of shaped like starfish that sit around the end pieces of salivary glands and sweat glands. And although they are epithelial cells, they have the contractile ability of smooth muscle, so that when they contract, they squeeze free-form secretion out of the duct system and onto a surface. The most obvious place where these cells function is in the phenomenon called milk letdown. You know that many women who are nursing have babies that are, the baby cries because it's hungry, and they suddenly start leaking milk from the breast. And that's due to the fact that the myoepithelial cells have undergone a conditioned reflex, and oxytocin from the pituitary gland causes these cells to contract. Somebody got very smart idea. They realized that it was causing that if oxytocin was causing myoepithelial contraction, maybe it also causes contraction of smooth muscle. Well, when this was examined, it turns out that oxytocin plays a major part in giving birth. That is, when a fetus is in the right position to leave the uterus, the pituitary will produce oxytocin, which causes strong contractions of the smooth muscle. Nowadays, it is used clinically so that when an obstetrician feels that the fetus is in the right position, head forward and so on, they will inject a woman with some oxytocin, and this damn near pops the baby out. It really does work. So it's an important hormone during the birth process. And as I said, it's now available in artificial form, and it's used very, very widely. But it all comes originally from the study of the pituitary gland. Okay, let's take a 10-minute break, and then we'll talk about the thyroid. Interesting gland in its own right. Okay. We'll be back by about 2 minutes to the hour.
So I'm just gonna check.
Curiosity that I
and I'll
meant
get
to
to
mention when we were talking about human growth hormone. Anybody have any idea what is the tallest true recording of the tallest person who ever lived? Not stuff in the mythology, but I mean where there are existing medical records. Have any idea how tall such an individual was?
86.
Well,
No,
I will tell you, he was about 8'7". 8'8". That's pretty big. Now, you know, there's a major problem being that human heart was not designed to pump blood over such great distances. And such individuals, these pituitary giants, all die in their 30s. And they're usually relatively crippled long before they die, simply because their muscles are not strong enough to support that skeletal structure that is responsible for their hugeness. Just a little bit of curiosity that I thought you might be interested in. Okay, let's move on then to the thyroid. Thyroid is an interesting organ. Have you seen the thyroid in gross anatomy? And you realize it's got two major lobes on the neck that are joined together by a kind of isthmus tissue. One examines these glands, it becomes obvious that they consist of hollow walls of cells. That epithelium is simple cuboidal epithelium, although under certain circumstances that epithelium may get very low, so that we can talk about low cuboidal epithelium. But it's a simple, one-cell thick structure. Such structures are referred to as follicles. So the thyroid consists of a series of follicles which are of different sizes, different diameters. They're all over the place. And they all contain an eosinophilic material, which is referred to simply as colloids. Okay, so highly vascular, and if you remember what the thyroid looks like in a fresh cadaver, it's very red because of the presence of a tremendous number of small blood vessels and filled with colloid. Now, thyroid follicles produce one hormone, thyroid hormone. And the way in which they do is very unusual. They engage in a kind of secretory cycle that is unmatched by any other cells in the body. If you examine the stimulated thyroid follicular cell, this is after it's been stimulated, you would see that the cytoplasm contains a lot of rough endoplasmic reticulum. And the rough endoplasmic reticulum produces a protein, which we call thyroglobulin. Thyroglobulin is a protein that is made in the RER of the follicular cells. This thyroglobulin is packaged in granules, which are then exocytosed into the lumen of a follicle. So colloid really consists of thyroglobulin. Okay, so far so good. Now, what happens is that these cells that make up the follicles are able to concentrate the element iodine to concentrations thousands of times higher than what is found in the blood. And this iodine is added to the thyroglobulin. And it's usually close to okay, so the thyroglobulin has become iodized around its periphery. Now what happens is if thyroid hormone is required, the cell sends out pseudopods and grabs hold of a chunk of thyroglobulin. It internalizes that thyroglobulin, and that body containing the thyroglobulin fuses with a lysosome. And a lysosome breaks down the thyroglobulin into its component amino acids. And the two major amino acids that are produced are thyroxine and another one, triiodothyronine. That's TRIO. Two different amino acids, both of which are iodized. And thyroxine has 4 iodides on it, and triiodothyronine has 3. For short, thyroxine is called T4, T3. Both of them, T4 and T3, are thyroid hormones. They both have the same function. They are released into the blood by exocytosis. That is, they move out through the bottom of the cells into the connective tissue and then diffuse into the numerous blood vessels that are found surrounding the epithelium. The T4 is quickly changed to T3, but both of these amino acids are thyroid hormone. I mean, is that clear? Let me repeat what happens again. When the follicular cells are stimulated, they make a protein, thyroglobulin, in their endoplasmic reticulum. The thyroglobulin enters the Golgi apparatus where it gets packaged into granules. The granules are exocytosed into the follicle so that the colloid consists of thyroglobulin. Thyroglobulin is not a hormone. It's just the beginnings of a hormone. Again, if thyroid hormone is required, the follicular cells will send pseudopods into the colloid, grab a chunk of it after it has been iodized, takes it into the cell, breaks it down by lysosomal activity, and two of the amino acids that made up that iodized thyroglobulin are thyroid hormones, T4 and T3. T3 is the more abundant one because T4 gets changed to T3 very quickly in the blood. If you get a report from a physician where they've done a thyroid test, the terms that they will use will be T3, T4. So just remember that these are different terms for thyroid hormone. And then this hormone gets released into the connective tissue, hence into the blood vessels, circulates throughout the body. Thyroid hormone has a number of effects in the body. Probably the most important one is that it tends to control the rates of metabolism in the body. It has other effects as well, which we need not get into. Now you can see that a key step in this whole process is the iodination of the colloid, of the thyroglobulin. That means there's got to be an adequate supply of iodine in the diet. If there is no iodine, what happens? Well, cannot make thyroid hormone. The pituitary recognizes that there's not enough thyroid hormone. So it stimulates the thyroid to go ahead and try to make it. Well, the thyroid then makes thyroglobulin, but it can't go any farther because there's no iodine present. So the same process is repeated. It makes more thyroglobulin and more thyroglobulin all the time without making the hormone. Well, if you keep adding thyroglobulin to the follicles, they're going to increase in size. And the thyroid will get bigger and bigger. And that is essentially what happens in the condition that is known as goiter. Goiter is due to a deficiency very common condition, especially in Europe. If you go to the museum here, the art museum, and look at paintings, usually of royalty or very rich people from back in the 1600s, even 1500s, you'll notice that almost all of them are wearing these collars that are rough, you know, those collars that have vertical folds. It's not a stylistic thing. That was all done to hide goiters. Practically everybody had them. And the people who could afford it bought clothing that would hide it. But like every other fashion statement that sort of spread farther out from areas where iodine was in short supply, places where people had normal thyroid glands, but they liked those growths. But those costumes are strictly the result of there not being enough iodine in the diet. This used to be the goiter belt of the United States. It used to run through Pennsylvania, through Ohio, through Indiana, and so on. Why? Because fresh fish from the ocean was not available readily, and that was a major source for iodine back in the old days. Then they started adding iodine salt, and when you buy table salt, it's iodized on it, and the amount of iodine that's present is tiny, but it's quite enough to take care of your thyroid needs. Okay, so the thyroid is peculiar in its secretory cycle. It differs from any other secretory cell in the body. Again, repeat, thyroglobulin is made in the RER, is exocytosed by Golgi and secretory granules into the interior of the follicle. If iodine is present, the periphery of the thyroglobulin gets iodized, and then the cell reaches out, takes a chunk of this thyroglobulin into the cell itself, breaks down the thyroglobulin by lysosomal activity, releases these two iodized amino acids, which are thyroid hormone. No other cell in the body does this sort of thing that the thyroid does. Seems sort of wasteful in a way. That's evolution for you. Anyway, that's basically the story on the thyroid, except there's one other detail we should mention. Now, if one examines in the microscope, a light microscope, a slide of the thyroid, you will find cells that sit on the outside of the follicle. These cells are within the basement membrane of the epithelium, but they do not reach the lumen. These cells produce a hormone also. And this hormone has nothing to do with thyroid hormone. What they produce is a hormone called thyroid calcitonin. And we now sort of just drop the thyroid part of it, just call it calcitonin. sit on the outside of the follicle. They're known as parafollicular cells, para meaning near to, parafollicular cells, or calcitonin cells, or for short, just simply C cells. Now, what does calcitonin do? It's an important, hormone, and it is related to calcium levels in the blood. Now, you undoubtedly are aware that we require our calcium levels in our bodies to stay at a certain, very well-defined level. Too much is bad, too little is bad. Well, calcitonin is related in regulating calcium in the body. Now, if the calcium level is too high, calcitonin will, let me see if I got this right. Yeah, if it's too high, it will regulate the function of cells that make bone, osteoclasts. And these cells will take some of the calcium out of the blood and add it to the bone to be stored so that it cannot interfere with physiological functions. Okay. Next time when we talk about the parathyroid gland, I will tell you about some cells in the parathyroid that have an antagonistic effect that release calcium by turning on osteoclasts and helping to raise the amount of calcium. So our calcium levels are constantly being adjusted by two antagonistic hormones. There's calcitonin in the thyroid and parathyroid hormone in the parathyroid gland. And it's sort of a kind of push-pull technique. When you're using a microscope, for example, when you're looking through it and you have your hand on the microscope, you start turning out of focus, and you may be a little bit out of focus, high, low, and then eventually you get it just right. So these two hormones work against one another, and finally will regulate our calcium level to exactly the right level for physiological purposes. So thyroid then, as we call it, thyroid hormone and thyrocalcitonin, simply calcitonin. Again, in the microscope, C cells are not easy to find. We used to have a lab. Students used to go nuts trying to find them, but they're there. Okay, are there any questions about either of these two subjects? Thyroid? No? Okay, then let me remind you again that our next meeting will be at 8 o'clock, at 9 o'clock rather than 8. Talk about the renal gland and the endocrine pancreas, the islets of Langerhans. You've studied a lot about oil later.